Hello, guys and girls. The program you are about to hear will be both fun and educational, but it is not a substitute for medical advice. Although we are doctors, we are not your doctors.
Hello and welcome to Travel Medicine. As always, I'm your friendly neighborhood internal medicine doc, Dr. J.
Heyo, this is your amazing pediatric infectious disease doc and researcher, Dr. Santhosh.
Santhosh, I've had a song stuck in my head
Oh
all
no,
day.
don't.
No, you'll like
What?
it because it ties into your culture. Well, like not
Oh,
you specifically, but your people's.
by people's, yeah, I would strongly advise for the sake of political correctness and not losing, I don't know how many viewers.
No, no. Well, first off,
Yeah.
we're not losing any viewers because this is an audio show.
What? You can view our waveform on YouTube.
No, I promise this ties in today's topic.
Go. Okay. Okay.
Okay, so you know that one Bhangra song that was played in like every club through the early 2000s? It was remixed with Jay-Z, the da 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 da
Yeah,
da da.
yeah.
Meet
And the
me.
night,
Yeah,
yeah.
yeah, yeah. It goes like, meet
Yeah.
me at the pool around 7:59. Da 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 da
No,
da 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 da.
I don't know the Hindi lyrics, but that's not it. Or the Punjabi lyrics. Meet me at the pool. That's it.
Bring a noodle and a drink with the lime. And a hot
That's it.
chicken. Anyway,
I
um,
can't listen to that song anymore. Like forever.
so it got me thinking about a lot of other Indian adjacent things, and you're going to love this. It's a classic Dr. J non sequitur transition.
Sure.
So we go from Bhangra to Bollywood to India
Mm.
to lassi, a fantastic Indian drink.
Oh, okay. That's the, you're we're I'm following with you. I'm on the train. I'm on the train.
Okay, we're about to derail the train because today we're going to be talking about lassi to Lassa.
Oh.
That's right, it's time for another Around the World in 80 Plagues.
You just went from India to Africa, just like thousands of miles west.
And only
not,
one vowel.
not that there aren't Indians in sub-Saharan Africa. There are lots and lots of Indians in sub-Saharan Africa. Uh, yeah. So,
So,
okay. So you want to go from some nice, tasty yogurt mango treat to bloody diarrhea and death.
I was thinking more just hemorrhagic fever, but if you want to focus on the bodily fluids aspect of it, sure.
Well, yeah, I mean, you're going to bleed from everywhere. Just, by the way,
Yeah.
I'm not implicating that Lassi causes Lassa.
No, the two are wholly unrelated, aside from the
Completely.
fact that this is what led me to research this episode. One Punjabi song, and next thing you know, I've written an entire episode about a fever in Africa. So,
I need
welcome to my
your.
brain.
We're gonna. There's going to be anthropologists this thousands of years from now studying your brain in a jar like they have with Einstein.
Do you know, incidentally, human brains, like
Yeah.
preserved human brains, there
Yeah.
is a surplus of them. Like everybody thinks they're rare, they're not. They are
They're not. No.
all over there are so many human brains just casually laying around
Well,
for
not
anthropologists
casually laying
by
around.
no, by archaeological standards, pretty
Yeah.
casually just laying around, littering the globe.
Yeah.
Something
Oh.
like 4,000. But that's
Yeah.
another episode. Let's get into this 80 plagues.
Yes, please.
We are going to hop in our Wayback Machine, head all the way to January of 1969, a little bit before I got my first real six string, which I
Oh,
bought at the Five and Dime.
no, very nice. I thought you were going to talk about how you were going to party like it's 1969, but that's the wrong year. That's not the one that Prince sang about.
No, you see, we have it's 1969,
Mm-hmm.
and we do have a retired Chicago nurse Laura Wine.
Oh, okay. That's not her name.
That is her name.
Oh,
Uh,
okay.
at that time, in charge of the obstetrics unit at Lassa General Hospital, who
Oh, it's a region.
had just ended a vacation back home in the United States, and on the way back, returning to Nigeria, Lassa,
Mhm.
Nigeria, she made a brief stop in Sierra Leone.
Oh, beautiful. Yeah, just one of it's very poverty stricken, but in terms of the area and the peoples, it's one of the most beautiful places on earth.
Appropriate to one of our other recent episodes, she was a missionary nurse affiliated with the Church of the Brethren,
Oh, okay.
and she happened to notice as she returned home to Nigeria that she was running a touch of fever.
Oh, uh-oh. Okay. So, fever in a traveler. By and
hmm
large, we think of malaria or salmonella, like typhoid. Okay.
All things for which there were treatments available in the 60s.
Mm-hmm.
However, she continued to get worse. The fever continued to climb and spike. All treatments were of no avail, and soon she died. At which
Oh no.
point, it became clear that this disease was something new, not seen before, and obviously highly transmissible, as not long after, a doctor and nurse that had treated her in Sierra Leone in a town called Jos also fell ill and died.
Oh, no. And I'm guessing that the poor doctor and nurse did not wear proper uh, protective equipment or PPE.
Well, as we covered in our medical missions episode just a few weeks back, there's not a lot of it to be had, even when you are aware of sterile technique. It is not always able to be practiced.
Sure.
But when a third nurse named Penny Pineo, who had cared for another case, fell ill with a spiking fever, chills, profound weakness, and an ulcerated throat, she was evacuated to New York Presbyterian Hospital.
Mm. Okay.
At the hospital, virologist Jordi Casals Ariet and his
Okay.
team at the Yale University Arbovirus Research Unit
Hmm.
looked at blood samples from the nurse in search of what they suspected was a brand new virus.
Oh, okay. Okay. They were hunting down something novel. This didn't look like anything that they had seen before.
They did manage to isolate a single stranded RNA virus in what
Yes.
was a brand new family, arenaviruses, which I'll have you talk about in just a moment.
Yeah.
And they then named that virus for the village where it was discovered.
Oh, okay. So the town of Lassa.
Yes, but remember,
Okay.
our first case had been traveling to Lhasa, even though it was
Oh.
discovered. But our virologist, Dr. Casals Ariet, became infected while handling the samples.
Oh, oh no. Oh no, now we've got laboratory transmission. This can get ugly fast.
And it did, as he almost died, being saved only by a very risky transfusion of antibodies from our nurse Penny Pineo's blood, who had had a devastating and prolonged illness, but managed to recover. So he's like, well, we know you survived. Let's just grab some of your blood and take it.
Okay, so they used what's called hyperimmune or convalescent plasma, meaning that a person has been exposed to a certain disease, recovered, they have formed humoral immunity, meaning that they formed antibodies. So you just grab it. You don't care what antibodies are in here. You just purify out the antibodies and you give it as a transfusion. We have been using this actually as treatment for diseases such as COVID, and there's one form of it for malaria. So this does work in theory for a host of different viral infections.
Well, in this case, it worked in practice. But when
Hmm.
Casal Ariet's technician, Juan Roman, also became infected and died,
Oh no. Oh no. Oh.
Yale immediately halted work with the live virus and
Yeah.
transferred it to the CDC's hot lab for the hot new diseases. Oh my
no,
god, Lassa, it's so hot right now.
no, no. It's the hot lab, so called because these are the most, important, scary pathogens. It's also damn hot inside of the BSL-3 suit that you have to wear in order to handle these things.
I don't want to get Lassa, but this suit is so hot right now.
No, no. Well, if you get hot inside of the suit, you're less likely to get the Lassa.
So, results from the CDC
Yeah.
indicated that this was a previously unknown arenavirus, which has also been observed in several other African countries and has claimed many victims and
Mm.
ultimately became known as Lassa fever, named by the US CDC in the early 1970s. So we will follow up with that unfortunate naming choice in a moment. But what can you tell us about arena viruses?
Yeah, so arena viruses are all what we call ambisense RNA viruses. So they can be sense, meaning that like the DNA material, or sorry, the genetic material can be read immediately once it invades a host cell and it can replicate, or it can be antisense, meaning that you have to make a copy of the RNA to the sense side and then copy it. The They're so frustratingly weird because it's a very, very small genome. And, you look inside of its capsule, you cut it open, and inside it looks like our little arena. You have the ribosomes of the virus that actually it stole from the host cells. And you have, all the other RNA particles in there. And so it was named arena because arena means sand. So when you think of the Roman Colosseum or the arena, they're talking about the sandy bottom on there. The arena viruses have a bunch of different genera. So Lassa is part of them. But there are tons and tons of other viruses under here that infect mostly rodents. But every now and again, they make the jump over to humans and they cause diseases like this one, which is we're going to be talking about hemorrhagic fever. We've talked about all the deaths so far. We haven't told you how they died, which is rather gruesome.
You dirty rat.
Yeah, they are dirty rats. Yeah, absolutely. it's
Well,
it's all rodent
before
transmitted disease. Yeah.
before we get to how it kills people, we're going to
Yeah.
get a little meta and talk about how it almost killed a town.
Oh, yes. Oh, I mean, a town full of people.
No, I mean the town. Uh,
Oh, okay.
because giving this new arena virus the name Lassa
Which is
had
not where it came from.
had pretty severe consequences for the town of Lassa.
Oh no.
It
Oh no.
rapidly became a wasteland. Physicians began to decline transfers there. They didn't want to get infected with this new highly contagious virus they thought was located.
Oh.
Travelers stopped coming, and it wasn't exactly a hot tourist destination even before, and even missionaries were apprehensive to work there.
Oh no. Oh no. So whether you're trying to get people healthy or spreading the gospel doesn't matter. That's where the bad scary disease came from because that's the name of it. This is the same nonsense.
In an attempt at damage control,
Okay.
the Nigerian government sent a representative to the Health Organization to
Okay.
argue against implicating Lassa in the damning nomenclature. We don't want to be named for this. But can you imagine if instead of COVID, we called it, Cleveland 19?
Well, so there was for a while until we discovered that it was a coronavirus and something akin to the past SARS viruses. There was a thought of naming it after Wuhan, and that I think likewise would have been a very very bad idea.
So this Nigerian physician went to the WHO and said, "Hey, would you do us a favor and not name this terribly infectious, damning disease after our town? That'd be great."
And
Um,
hold on, the WHO being a stand-up, rational scientific organization said, "Of course, good sir, we will henceforth change it right away."
To which the WHO replied, "Oh,
No,
well, we'd
no.
really like to,
Oh
but
no. Oh
I
no.
mean, it's already in the literature, and once
Oh.
an organism is named in the literature, it's really difficult. There's a ton of paperwork,
Oh.
but I tell you what,
Oh.
the assembly agreed it is in fact stigmatizing to name
Yeah.
infectious agents after locations.
Okay.
So going forward, not counting Lassa or Ebola in Zaire or West
Mhm.
Nile, but after all of those, we will
Okay.
definitely stop naming diseases after locations.
Yes.
So go home and feel better about yourself. Oh, you come from that Lassa region.
Oh
Good luck
dear.
with that.
We could have named it, Castle's disease, Dr. Castle's. You could have named it Wine's disease after Miss Wine. No?
And meanwhile, Dr. Casales is like, "Oh, name this after me. I don't want anything to do with this."
Oh my god. That's heartbreaking, but okay, so we understand what it is from an epidemiological standpoint and how it's related to other, small RNA viruses that go from rodents to humans.
Let's talk about now that we have implicated the town.
Yeah.
Let's talk about how we found the original reservoir, because you have mentioned a couple times that it's rats, but it's not all rats. Come on, Santhosh.
No,
Not
it's not
all not
hashtag all
all
rats.
rats.
It is not hashtag all rats. Yeah.
So, 1970, this disease gets named Lassa fever.
Yeah.
1972,
Hmm.
during a Lassa fever outbreak, stigmatizing, but don't worry about it. We'll take care of that.
Sure.
During a Lassa fever outbreak again in Sierra Leone, Thomas Monath, a virus hunter at the CDC, set out to find the animal reservoir. Now, having worked for the CDC, he knew that a lot of these viruses are spread by small vertebrates.
Yep.
So, in a highly dangerous and made for TV adventure, I'm thinking something like
Oh,
Crocodile Hunter, they
yeah.
trapped
Oh
they
no.
trapped over 641 animals and removed tissue and blood samples. Bats, shrews, monkeys, and a single turtle turned up nothing. But
Okay, so
eventually,
uh-huh.
he did isolate the virus from 14 different specimens of the multi-mammoth mouse Mastomys natalensis, and it's called that because of its long rows of nipples for nursing great big litters.
Yeah. So, in fact, under arenaviruses, these are called mama arenaviruses. So, the species is Lassa mama arenavirus. Literally,
Lots of
the
marina virus coming at you now.
well, if you say arenavirus, this Josh is the sandy boobs virus from Lhasa. Yeah, that's the full Latin translation.
But when the paved road ends,
Yeah, yeah.
you know you are entering a Lhasa community
Sure.
in West African nations such as Sierra Leone,
Mhm.
Liberia, Guinea, and Nigeria,
Yeah.
which, not coincidentally, now holds the region's foremost research institute for viral hemorrhagic fevers and emergent pathogens.
Wow. Okay, so not just Lassa, but all other related and unrelated hemorrhagic fevers, including Onyonyo and Ebola and all these.
Yeah, I guess Nigeria figured, listen, if you're going to start stigmatizing us with towns, we might as well also be known where for this institute that studies all of these diseases. It's
Yeah.
kind of a rolling with the punches.
It's a really good idea. The disease is here. Let's learn about it here. Absolutely.
So, our Mastomys natalensis, multi-mammoth rats,
Hmm.
are the most common rodent in tropical Africa. They are found predominantly in rural areas, but in dwellings more than in the surrounding countryside. They like to hang out in the huts and the houses. Members of the genus are infected persistently They shed the virus pretty much nonstop in all their excretions, and humans are infected by contact with the rats or their excretions or by eating them, as they are considered a delicacy and eaten by up to 90% of people in some regions of West Africa.
Okay, so it's a local staple.
Yeah. And
Okay.
the ones found, now again, remember I said they're found in dwellings. Rats found in houses of infected people are seropositive for the virus 10 times more often than those in control houses or the surrounding countryside.
Got it. Okay. So seropositivity follows the animals.
Yeah, and there will be
Okay.
signs as you leave the paved road. Do not touch or play with rats. Do not eat food contaminated by rat urine or excrement. Take precautions to prevent rat bites and scratches. I feel like some of those should be self evident.
But at the same time, if you are living in an endemic area where these animals are everywhere, and I say this to even my trainees in infectious diseases, the diseases don't come around and infect us. We live in their world and we happen to be in the friggin' way. So that means that if we don't want to get sick, we have to be the ones who make sure not to step in it.
How dangerous is it? Well, it causes tens of thousands of deaths every year, with about 5,000 deaths from every 100 to 300,000 infections, which means in areas of Sierra Leone and Liberia, about 10 to 15% of people admitted to hospitals every year are just guaranteed to have Lassa fever.
Yeah, this is a little bit like the flu of this area. You can think of it that way, which is a scary ass flu to have, but that's what it is.
I'm so glad you mentioned that, Santosh, because in
Yeah,
Nigeria, annual outbreaks occur every year from December to March. They
there you go.
have hemorrhagic flu season.
They do. They absolutely do. And I'm guessing that's the time when the animals proliferate and they're much more abundant and, mingling or commingling with humans much more than the other months of the year.
Either the rainy season, which scares more of the rats indoors,
Hmm.
or it is the not rainy season when they are more able to breed. It's one of those.
It's a flip of a coin, sure.
But there is something with the climate that affects these outbreaks.
Ooh, I found it, Josh. It is the dry season, December to March.
Okay.
So the wet season keeps the rats away, May to November.
Okay.
Yeah.
So why do
Oh,
you
with a little caveat. Our most recent data from Kenema showed that admissions were highest actually during just the change from the dry to the wet season. So like March, April, something like that.
So, why don't we talk about Lassa in terms of its symptoms and how it is similar and different from Ebola, which is probably the one that most people hold in their heads as the example of a hemorrhagic disease?
Yeah. So when a person acquires Lassa, it's pretty much indistinguishable at the start. Anywhere from 6 to 21 days before, they will have had some sort of contact with rat or rat fluids, and you're gonna have a high fever. You'll try antimalarials because that's still one of the most common fever diseases, antibiotics. None of these will work. And then you talked about a little of these in the historical cases, Josh. Aside from the fever, you'll have a sore throat or pharyngitis, pain behind your chest or your breastplate, which is called retrosternal pain. And then if you continue to watch and do laboratories, you'll get proteinuria. Um, which means you'll spill protein in your urine as it attacks your kidneys. Then, if you go through, to stage 3 after about 7 days, now it'll start attacking and destroying your ability to actually clot, etc. And you'll have the mucosal bleeding, so bleeding from your mouth, your nose, your eyes, internal bleeding, which you can't see. And eventually it'll turn into confusion, disorientation, and it turns eventually into an encephalitis. you'll lose your hearing, small percent of the time. And, people will die either from an internal bleed somewhere or from just overwhelming kind of brain edema and inflammation.
About 1/4 of survivors become deaf, although half of those will regain some hearing. And the
Yeah.
earlier this is successfully diagnosed, means the likelihood of recovering becomes much higher, and we'll get into treatments for that in a moment.
Hmm.
Uh, Now, Lassa is less contagious and less lethal than Ebola, and around 80% of infected people in Africa have no symptoms, and those who do have mild symptoms.
Yeah.
That would not be true for someone like you or I who does not have the generations of adaptive mechanisms to make this
Yes.
slightly less lethal.
Yeah.
On the basis of a case series of 140 men and 128 women admitted in the town of Kenema during 2001, we learned that the duration of illness can vary. The most common interval between onset of symptoms and discharge from the hospital was 17 days. So, about half of the patients admitted with known Lassa fever were in the hospital for at least 10 days. And if they made it 10 days, things were looking pretty good because the time between onset of symptoms and death was 5 days. Half of those
Yes.
dying within 2 days of admission.
Yeah, yeah, absolutely. Because most folks will not, they won't get a fever and come straight in to the hospital. They're going to wait until they have telltale sign symptoms or they're bad enough clinically that they can't stay home. So catching a
Well,
person in the hospital,
it may also be that the nearest hospital could be miles upon
oh,
miles away and there
that's
is not
true.
reliable public transportation. You may not have the money to be admitted. Remember, we're
That's
talking
true.
about a disease predominantly endemic in a developing nation that is resource poor.
Yes.
Uh,
Nations, yeah, all along the west of Africa there. Yeah, absolutely.
So, and a lot of these deaths were resulted from patients not receiving our one of our few treatments, ribavirin, within the critical first 6 days of infection. So
Yeah.
now you're a pediatrician, Santosh. Lassa is particularly severe in pregnant women in the third trimester.
Yeah,
Uh,
as is most diseases, a woman who is pregnant in the third trimester has a functionally suppressed immune system so that her immune system doesn't attack the baby, which is slowly but surely differentiating itself from mom. So for a while, she has a foreign body in there. So she's functionally immune suppressed. And like with many other diseases, she's much more vulnerable to severe disease from Lassa when she's in that phase of pregnancy than at any other time in her life.
Now, symptoms in children are similar to those in adults, but infant infection can result in something called swollen baby syndrome,
Yeah.
which is
Yeah.
exactly what it sounds like. They become swollen with fluid like edema, abdominal distension, bleeding, and ultimately death. So, when Lassa infects women in their third trimester, you have to induce delivery for the mother to survive. This is because, as you mentioned, the virus has an affinity for the placenta and other highly vascular tissues. Now, no matter what course of action is taken, meaning you give drugs in time, you don't give drugs in time, the fetus has only a 1 in 10 chance of survival. So the focus is always on saving the life of a pregnant mother with Lassa.
Right.
Following delivery, women then get the same treatment as other people with Lassa.
Yeah. And there is that in terms of if baby has to come early, if you're very early in the third trimester and baby isn't viable at that time, or passes away from being very premature at this time in medical science, we don't have a good way to reconcile this, especially in a resource poor area. You save mom, and then okay, then after that, if you can save her, now she's immune effectively. She has antibodies, and this isn't a danger any longer. And if she wants to have another baby, if she can have another baby, then it's an option. If mom dies, almost certainly baby's going to die with her, and you've lost two.
So how do you diagnose this?
So it's not a wonderful way. I hate anything where you have to look for the smoke and not the fire. And we're still kind of stuck in that era, Josh. So, yeah, we use antibody testing really more than anything else. So you can actually test for the antigen and the antibodies against the virus using a immunosorbent assay or an ELISA. And it's pretty sensitive and specific. It's not fantastic, but you do have to know what you're looking for. If you're in Sierra Leone and other adjacent countries, of course, this is high on your differential diagnosis of a person coming in with fever and sickness. And so you're going to check for it. Here in the United States, finding testing for Lassa is few and far between and difficult unless you're like where I work, which is called a special pathogen center. And you have to find these antibody kits with a public health helper.
With reverse transcription polymerase chain reaction, or PCR, something I know you are intimately familiar with.
Yeah.
Lassa can be diagnosed in all patients as soon as the third day of illness.
Yes,
But
we have RT-PCR.
But unlike the ELISAs, which are about 88% sensitive and 90% specific, immunofluorescence in PCR identifies only about 50% of the patients, which in Africa is not a terrible coin flip because you can start treating on the assumption it is. Outside of endemic regions, 50%, maybe you have the disease, maybe you don't. Probably not the ideal diagnostic tool.
Yes, you don't want to jump to that. It is fast, I'll say. RT-PCR, if you have everything set up properly, is a very quick methodology. But you don't want to have to rely on something which gives you a coin flip of a chance of proper detection. The reason for this sounds a little bit weird on its surface, Josh. You actually don't get a massive viremia when you get sick with Lassa virus. So, it'll get into your body and invade cells. It starts replicating. But the problem really isn't that the virus itself is going and infecting, your tissues and destroying it just wholesale. The problem really is that you have hyperactivity of the immune system, and you have a hugely activated immune response, and these mechanisms which are actually supposed to protect us, which cause inflammation and are supposed to wipe out viruses and stuff, these are the things that are likewise responsible for causing these leaky capillaries, and you get edema everywhere. And then likewise, destruction of, your clotting system and interference with that so that you bleed. So you don't actually have a ton of virus going around, which is why RT-PCR isn't as, sensitive as you think it would be for a molecular test.
And we're going to get to the interesting ways that this disease avoids and confuses and tricks testing in just a moment. But once you have been diagnosed with it, once you are suspected and it's confirmed, then our treatments are limited but not non-existent. The main one right now is a drug called ribavirin, which began its life as a AIDS drug for HIV, another RNA virus.
It has an activity against a host of, RNA viruses. It is used occasionally to try and fight bad, RSV in an immunocompromised individual. It has activity against hepatitis B. So it's actually not a one-trick pony. It can attack a lot of RNA viruses. The problem is the efficacy itself isn't actually all that great.
Although, when again, you're in a resource poor area, some chance is better than none. Now, it is about twice as effective when given intravenously as when taken orally. But in both cases, even though it is, again, not the most ideal drug, if given within 6 days of the start of the illness, the start of symptoms, it can
Mhm.
reduce deaths by 90%.
Wow. That's some power.
But how often can you get to a hospital in West Africa within 6 days of the start of symptoms? Even if you
Yes.
are on the ball and say, you know what, I am feeling slightly altered, not myself, I better set off on the 5 day trek to the hospital.
Sure. Sure.
So, again, this is where the infrastructure really is almost as damaging as the illness itself. But let's say you do recover. Even
Mhm.
after recovery, the virus does in fact remain in bodily fluids, including semen. Um, we only learned this through some early study, but the years of civil unrest in Sierra Leone from pretty much the entire 90s halted the investigation and study of Lassa fever at a specialist unit in Sierra Leone. They were too busy fighting civil wars to be studying this disease. What they had
Yes.
learned before the Civil War started was the virus is excreted in semen for up to 3 months after infection. We do not know how frequently it may be transmitted through sexual intercourse. It is definitely a possibility, but beyond that, we really haven't been able to study.
Yeah, it is not classified right now as an STI. But the problem with hemorrhagic fevers as a general rule is pretty much every single tissue and fluid on and in the human is infected. And that's why this damn thing is so communicable. In the definitive host, in the rat, if it's going around, and just like you mentioned, Josh, if you are bitten, if you're peed on, if you're spat on, if you are bled on, anything at all, there's a very, very good chance of Lassa to transmit and start doing its work. So, yeah, you mentioned semen, but the problem is it's going to be in tears, it's going to be in saliva, it may be in sweat, and it's definitely intermittently in the bloodstream and in tissues if there's any, cuts or anything else like that. So there is it's a false sense of security when a person is perfectly healthy that maybe they get back to regular life and they're still hot. They're still transmitting. And a lot like a person who's recovering from COVID, you have to set a time of, oh, no, no, okay, you're still infectious. Please don't go around hugging and kissing everybody, please.
Unless you'd like people to start bleeding from orifices.
Yeah, that's true. That's true. It does depend on where you are here in Western Africa. As you mentioned, a lot of people, you have enough folks who get infected and recover from Lassa that you do have quite a percentage of people who are protected because they already have antibody on board.
Well,
Uh,
since you mentioned that,
oh, yeah, yeah.
researcher Pardis Sabeti has developed an innovative method to kind of look over the human genome for
Oh, okay.
signals of recent positive natural selection. And
Okay.
the single strongest signal turned up in the Yoruba people in Nigeria. They have an
Okay.
increased frequency of a variant in a gene called large. It's an anagram for something just large. Uh, the gene the gene encodes an enzyme that modifies a surface receptor that the Lassa virus uses to gain entry into cells. So, Sabeti and colleagues suspected that this variant may actually confer some protection against Lassa fever, if not enough to completely prevent it, enough more to get the mild form, but they didn't have enough sequence data to know for sure. And the reason,
Oh.
so what they then had to do in order, because again, the ultimate goal is to be able to produce a vaccine for Lassa. So another study started looking at the analysis of genetic variation among different Lassa viruses. And this was
Oh, okay.
published. So the first study I talked about was looking at genes in people who get
Yes.
infected with Lassa and may have some resistance.
Right.
But this study from the journal Cell in 2015 actually looked at the virus itself, and we found that it has some pretty ancient origins,
Okay.
emerging in present day Nigeria about 1,000 years ago, then traveling westward to Guinea and Liberia 400 years ago, and reaching Sierra Leone a mere 100 some odd years ago. And it is one of the most genetically diverse viruses known. 7 distinct lineages have been identified, each one confined to a specific geographic area. So that means the Lassa virus is a little bit like a Pokemon. It gets to an area, it then happily evolves and mutates in the rodent populations that don't mix. Eventually, it travels to a new area and it starts and it gets a whole brand new identity and personality while causing the same symptoms. So there
Yeah.
are 7 different lineages that we know of.
Yes. And under arenaviruses themselves, or even the mama arenaviruses, there, I think there's 30 plus species that are circulating their way through rats. We specifically get infected by a couple of them, such as Lassa and another fun one called lymphocytic choriomeningitis mammarinavirus. And so that's kind of the scary thing. RNA is extremely unstable and the rates of mutations are super fast. The number of deficient or kind of bad viruses that happens when it replicates is probably way, way more than the ones that are able to stick around. But that means that you select out all these dud viruses every time it replicates, and you're left with a bunch with, like, tiny little mutation here, mutation there, and boom, all of a sudden you were protected against Lassa, but you're not protected against that Lassa.
And that's where
Uh,
we get our most recent studies that take the genetics from people and the genetics from the virus and slam them together to see what happens.
okay.
And in a study published only in the last month or two in Nature Microbiology, Dr. Happy and colleagues looked at Sabetti's initial observation and report that the large variant is associated with a reduced likelihood of getting Lassa fever in a Nigerian cohort where the Yoruba people are located, but not with a Sierra Leone one, which means humans are also evolving genetic variants specific to their regional variant of Lassa. So they identified 2 different human genetic variants associated with Lassa fever fatality, and they're looking at other local tribes that have smaller nomadic ranges to see if they can identify genes unique to each Lassa variant for that region. That's a lot to digest and take in there. We're looking for Pokemon trainers to tame the specific Lassa Pokemon for an area.
Sure. If you can stop the flux of human beings and then the hosts, keep them endemic for some period of time, this is the natural host virus type of relationship over time that you'll come around to a very what do you call it, infectious particle that's not as severe, because you come together with this kind of like, all right, I'm not going to mess with you, you're not going to mess with me, I'm just going to show up, replicate for a bit, and then take off, which is the way that we think and we hope that, for instance, COVID is doing, also being a small, or rather a larger, but an RNA virus. So yeah, the problem that we have with this, I think the analysis is absolutely beautiful, but the major problem we have, Josh, is that people, animals, and viruses do move. And when you have things like famine and war, which push populations around and then, create a worse vacuum of medical care, then you're going to kind of give a perfect, what do you call it, storm for these viruses to mutate and become virulent again.
And speaking of perfect storms, remember global warming and climate change also play a role in this
Oh dear.
because by 2070 under a moderate climate change scenario,
Yeah, not the apocalyptic ones. The like, okay, this, yeah, okay,
just saying like, hey, let's say we actually do some of the things we said we're going to and we make these changes and we only increase the temperature by like half a degree instead of 2 under
sure.
all those moderate conditions. The area suitable for Lassa virus circulation could expand from just West Africa to Central and East Africa as well. Now, not all of it, but Lassa will become a much bigger issue across all of Africa instead of being limited to just one particular part.
Oh dear. And by the way, we are focusing in on this particular hemorrhagic fever. There be others. And these generalizations are the same for those when we're talking about epidemiologic spread under a changing climate model.
Which means we're going to need better drugs because more people are going to start getting this. And that is where we will kind of wrap up, where a new small molecule drug, favipiravir,
Mm-hmm.
which interferes with viral genome replication, has just finished a phase 2 trial. Now, it's already been licensed in Japan to treat influenza, and a drug that could treat influenza and bloody influenza
Yeah.
would be great. So far, vaccine makers have focused just on treating lineage 4 of Lassa, which is prevalent in Sierra Leone, Guinea, and Liberia, with the sincere hope, that the shots will be cross protective. They're also
Nice.
looking into messenger RNA vaccines, such as those we saw developed during the COVID era. Uh,
Yeah. If you find a stretch of RNA that can, using your cellular machinery, spit out a protein that is a protective antigen, right? That makes protective antibody. The beautiful thing about mRNA tech is that you can go straight back to the lab and change a little bit of the genetic code and give another shot. And you should be able to provide protection again if Lassa decides to mutate or a new strain comes in. So it's highly versatile. It has a lot of promise. It doesn't mean it's 100% that it's going to work and it's going to be wonderful, but it's much more flexible than making a single protein virus.
so, from small Nigerian town to potentially global hemorrhaging problem,
Yeah.
uh, we've gone on quite the journey. So, what's the
Yeah.
takeaway here? Don't eat rats, do not touch rats, do not play with rats, and avoid eating food contaminated by rat urine or excrement.
That's true. And if you're a healthcare worker, you want to be very, very well versed in PPE, protective equipment, so that you save yourself so that you can save your patient if you're in the midst of a hemorrhagic fever ward. That's actually some of the most precious, things that can be donated is PPE, in order to take care of the healthcare workers who are saving the lives of people with hemorrhagic fever over there. So yeah, and just don't take any chances, folks. Please be very careful when you're out there. If you decide to go over to Western Africa, I will still contend, beautiful area, wonderful people, lots to see and do. But from the standpoint of illness, you do have to take care of yourself. Remember what Dr. Josh said. You have not been co evolving with these viruses. Your chance for death is actually way higher than that of a native West African.
On that cheerful note,
Yeah,
that's it for this week and our Around the World in 80 Plagues, which I think is our first of the season? Second? I
it,
don't know.
it might be. Yeah, I'm a little surprised you didn't do the Bhangra music. Please don't do the Bhangra music.
So that's it for this week. As always, we love to hear your comments, questions, and feedback. If you'd like to support us spiritually, emotionally, or financially, links to do that are in the show notes, along with links for further reading. This show is produced by me with a lot of help from Dr. Santhosh and friends. Our theme music is composed by Rachel Leisure. And until next time, as always, keep a song in your heart, soap on your hands, a shot in your arm, a spin on your globe. And when you've done all of those things, pick a country that looks interesting to you, bring your proper protective equipment, and meet me at the pool around 7:59. And until next time,
I'm going to get all the hate mail.
And until next time, as always, happy travels.